Howdy, my name is Jazlyn Gonzalez. I am part of group number 6 with Andres Garza, and today we will be presenting over Lab 3 Culture Media. Culture media is essential in microbiology because it allows for the growth, isolation, and identification of specific bacteria species. Each media has specific purposes, some being selective and some being differential. For example, blood agar can show different patterns of hemolysis. McConkey agar can separate lactose fermenters from non-fermenters, and mannitol salt agar can tell apart different types of Staphylococcus. We also tested motility to see which bacteria can move and used thioglycolate medium and anaerobic chambers to study oxygen needs. By looking at colony growth, color changes, and where bacteria grew, we were able to learn how these tests can help identify both known and unknown organisms. Now for our materials. For media, we used blood agar, CNA blood agar, bioesculin agar, mannitol salt agar, eosin methylene blue agar, McConkey agar, Salmonella shigella agar, motility medium, and thioglycolate medium. For cultures, we use Alkylogenes faecalis, Clostridium perfringens, Enterococcus faecalis C, Escherichia coli, Klebsiella pneumoniae, Salmonella typhimurium, Shigella flexneri, Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus pyogenes, and our unknowns were number 136 and number 276. For equipment, we use an inoculating loop and needle, Petri dish racks, a test tube stand, a Bunsen burner, permanent marker for labeling, an incubator, an anaerobic chamber, and parafilm. Now for our methods. For the plates, we used a marker to divide each plate into labeled sections as followed below. Then we used an inoculating loop to inoculate the assigned microorganisms into their corresponding sections, followed by incubating all the plates at 37 degrees Celsius for 18 to 48 hours. For the motility test medium, we used an inoculating needle to stab the motility medium tubes with E. coli, K. pneumoniae, and unknowns number 136 and number 276. Then we inserted the needle straight in and pulled straight up to avoid slicing. Lastly, we incubated at 37 degrees Celsius and examined for turbidity throughout the medium. For the thioglycolate medium, we inoculated the tubes with E. coli, A. faecalis, C. perfringens, and our unknowns number 136 and number 276. We then incubated and observed growth distribution from the top, bottom, or throughout the tube. Lastly, for our error tolerance testing, we streaked E. coli, A. faecalis, and C. perfringens onto our blood agar plates. We placed one set in an anaerobic chamber and the other set in a regular incubator. We compared growth between aerobic and anaerobic conditions.
Moving on to our results and observations, you can see a quick picture here of all the different tests of the cultures that we performed during our experiment. For some of the first results and observations that we had, which was with blood agar TSP, we can see that on the far left-hand side with K. pneumonium, there was a small dark streak presentation that was on the left-hand side. And then as for S. pyogenes, there was no observable change. Now for S. aureus, we did see that there was a dark orange streaking presentation, but for E. faecalis, there was also a streaking presentation. However, it was clear. For unknown number 136, there was a dark brown streaking, and for unknown 276, there was also a dark brown streaking. However, it was just a little bit wider in width. So for testing with CNA agar, we had a total of 5. Starting off with E. coli, there was a visible orange streak, and for S. pyogenes and S. aureus, there was no observable change. However, for unknown 136 and unknown 276, there was. The only difference was that for 136, there was a complete brown streak, and as for unknown 276, there was only a partial brown streak, and this was most likely due to an inefficient inoculation. As for bioesculin, our testing, we noticed that on S. aureus, there was no observable change. However, for E. faecalis, there was a hydrolyzed esculin, which resulted in a darkish brown state. S. pyrogens, there was also no observable change. However, for For unknown 136, we noticed that there was hydrolyzed echelon resulting in a darkish brown stain. And the staining also transferred over to the other side of unknown 276. And this was we're suspecting that this might have been due to some type of inefficient inoculation or a type of fall that might have spread the bacteria over. For unknown 276, we did notice that there is some staining, however, it is light in color. For our results and observations with mannitol salt agar, we noticed that on S. aureus there was a yellow pigmentation around our streaking, which has indicated the presence of fermentation. As for S. epidermidis, there was no visible streak, and the same for S. pyrogenes as well. So for unknown 36, there also was a visible yellow outline from our streak, which indicated fermentation as well. And for unknown 276, there was no observable change. As for our McConkey agar, we did see that there was no observable change for E. coli, and for S. typhimurium, there was no yellow outline. However, there was a clear streaking presentation. And as for S. aureus, there was no change. Moving on to unknown 136, there was no observable change. However, for unknown 276, there was a light yellow streaking presentation. Moving on to our results and observations for EMB agar, we noticed that on E. coli, there was no visibility, which in this case there was supposed to be. However, we're suspecting that this might have been due to some kind of possible error, since there was supposed to have been a green streaking presentation based off of our observations from other cultured bacteria. So for K. pneumoniae, there was a purple streak presentation, and for S. typhimurium, there was a visible streak. As for unknown 136, there was no growth. However, for unknown 276, there was a light brown presentation streak on the culture. As for SS agar, on E. coli, there was no growth, and the same for S. flexneri. However, on S. typhimurium, there was a clear yellow streak, which did indicate the presence of hydrogen sulfide being produced. As for unknown 136, there was no growth, but on unknown 276, there was a visible growth, which presented in a clear yellow streak.
So now for our results and observations. Under blood agar aerobic testing, we can see that E. coli shows solid streak growth. It is, however, a little bit blurry. A. faecalis does show solid streak growth. C. prefrigens shows little to no growth. There is one small speck that does show hemolysis. However, I was not 100% positive if it came from C. prefrigens itself, or it could have been a drop that was made during inoculating. For unknown 136, we can see that there was growth. However, it does not seem to be very prominent like the others. And for our unknown number 276, we can see that there was very clear growth. There was also a small dropping that made it down the middle of the plate, and that small growth does show hemolysis. Now for anaerobic testing, we can see that E. coli shows clear streak growth. It is also a little bit blurry on this plate, but it does seem to be more prominent than on the aerobic testing. A. faecalis did show visible growth, but not as prominent compared to when under aerobic conditions. C. prefrigens did show visible growth when put under light, and there was a clear zone surrounding the streak, which suggested hemolysis. Our unknown 136 under anaerobic testing did show better visible growth compared to the aerobic testing results. And our unknown 276 did show growth under anaerobic conditions, but there is still better visible growth in aerobic conditions, as under the anaerobic conditions, it's a little bit less prominent. Now for our results and observations of our motility medium. In E. coli, there was no visible growth along the stab line or in the surrounding medium, which led to the conclusion of this test being inconclusive. For K. pneumonia, there was growth seen close to the stab line with slight feathering, the possible outcome of a wiggly stab or heavy inoculum, and a thin surface film, no diffusing into the medium. This would suggest negative motility. For unknown number 136, there was no visible growth along the stab line or in the medium. This would suggest the inconclusive test results. And for our unknown number 276, the medium appears to be cloudy. There is turbidity throughout the tube. Growth is not limited to a stab line. This suggests that the organism is motile, making the test motility positive. The reason for our E. coli and unknown number 136 tests being inconclusive could have been most likely due to the needle being too hot when inoculating, the culture not being fresh or viable, or the medium not being made properly. Now for our thioglycolate medium. E. coli did show growth at the bottom of the tube, which indicates anaerobic growth, although E. coli is normally a facultative anaerobic organism. This test showed preference for low oxygen conditions, which could have been caused by excess inoculum deposited at the bottom and lacking even spread. The tube with A. faecalis did show a slight reddish color change and cloudiness at the top of the tube when put in front of lighting. A. faecalis normally grows in the presence of oxygen, making it an obligate aerobic organism, which was seen consistently in our results. C. perfringens showed visible growth observed in the middle of the tube with a small cloud of growth towards the bottom. C. perfringens grows best without oxygen, consistent with being an obligate anaerobic organism. Its distribution shows that this anaerobe thrives in little to no oxygen. Our unknown 136 growth was absorbed only at the bottom of the tube with the rest of the medium remaining clear. This growth pattern indicates an obligate anaerobic organism, since the organism grew only where there was no oxygen. Based on comparison with our controls, this result is most consistent with C. perfringens. Now for our unknown number 276, there was slight turbidity observed throughout the medium, with slightly heavier growth at the top, looking almost like a small thin sheet of film. This growth pattern indicates that this unknown is a facultative anaerobic organism, which is usually consistent with E. coli.
So, in conclusion, this lab media experiment demonstrated how selective and differential medias were able to enable microbial identification. Additionally, by combining colony morphology, hemolysis, motility, and aerotolerance testing, we were able to develop the profiles for almost each of our organisms. And additionally, it should also be taken into account that this experiment can be very tedious and also time consuming. So paying attention to detail is extremely important in order to achieve the most effective results. And thank you guys for listening.